Welcome students to another upload, and in this video I want to discuss pharmacokinetic analysis through physiology-based pharmacokinetic modeling as a potential area of research during M.Phill or Ph.D. studies. Before I proceed any further, here is my contact ID. Interested students could contact me for further guidance and assistance. Physiology-based pharmacokinetic modeling is a mathematical modeling technique for predicting the absorption, distribution, metabolism, and excretion of synthetic or natural chemical substances in humans and other animal species. PBPK modeling is used in pharmaceutical research and drug development, and in healthcare risk assessments as well, for designing optimized dosage regimen, prediction of toxic concentrations, adverse effects, drug interactions, and many more. Okay, now this is a very classic example of or symbolic representation of a physiological model, and you can see here we can see various organs in the form of a compartment, and these compartments or these organs are basically getting flushed with the blood through arteries and then getting emptied through veins. And Q1, Q2, Q3, Q4, Q5 is nothing, simply it is blood flow. You can see here down below. And CV1, CV2, CV3, CV4, CV5 are basically, CV is a representation of venous drug concentration, while CA is arterial drug concentration. So for example, it is oral administration, it is being administered, it is going to or coming to the GI lumen, then it would be reaching out to various organs through the blood flow. So this is a very basic and classic representation of a physiological model. Since PBPK models explicitly consider different organs and tissues, it is possible to obtain the quantitative characterization of concentration time profiles in the respective compartments. So such prediction is of high pharmacological relevance since it enables the estimation of drug exposure not only in the plasma but also at the site of action, means in various compartments, which are the organs basically, which may be difficult or impossible to measure experimentally. There are various commercial softwares which are used for pharmacokinetic modeling. Important one are SimSafe, PKSim, Physiology PK or PhysPK. The ones which I use are PKSim and PhysPK. So what is PhysPK? Disruptive software for modeling, simulation, optimization, estimation, and validation of physiologically based pharmacokinetic parameters in individuals and populations. PBPK company is working with many institutes and hospitals in collaboration as partners, for example, NRG, Netherlands Cancer Institute, PharmaMar, and many others.
PhysPK is a revolutionary software program for modeling, simulating, optimizing, estimating, and validating physiologically based pharmacokinetic parameters for individuals and populations. The PhysPK platform is a first-class simulation tool for modeling continuous discrete systems designed by Empresarios Agrupados International. It uses a non-algorithmic, object-oriented modeling language that supports multi-scale and strong models, both of which are reusable. It's based on physiology libraries that can include an object editor to modify the data of the components. The user drags and drops components from the palette onto the canvas, and then connects them and sets the attributes for each instance of the object.
Pharmacokinetic modeling has not only time and resource effective approach, but ethical considerations are also involved. For example, look at their 3R perspective. First R is for replace. Replace animal studies with other methods. Second R is for reduce. As many trials as required, as few as possible. Third R is for refine. Minimize stress of study animals. This chart is explaining role of pharmacokinetic modeling at various stages of medicine development. The second software which I use is PKSim Mobile Computational System Biology Software Suite. Together it is called. And PKSim is a comprehensive software tool for whole body physiology based pharmacokinetic modeling. It enables rapid access to all relevant anatomical and physiological parameters for humans, and the most common laboratory animals, mouse, rat, mini pig, dog, and monkey, that are contained in the integrated databases of the software. Now on to the building blocks of a PBPK model. So first of all, the thing which is required is the organism properties in the form of its anatomy and physiology, like organ volumes, surface areas, tissue composition, blood flow rate, and expression levels. All this information is already present in the database of the software, and you don't need to do anything. The thing which is required by you to as input information is the physicochemical properties and the biological properties of the drug of your choice or interest, which you want to investigate for pharmacokinetic analysis. For example, lipophilicity, log P value, molecular weight, pKa value. Similarly, unbound fraction, partition coefficient, active processes like Km, Vmax, KdF, these are known in the literature. You can add these values as well. Along with this information, the other information needed is formulation properties like the dissolution, function, and etc. Administration protocol, and the special events could also be added. All this information is then integrated by the software, and the results are generated in the form of pharmacokinetic parameters and plasma drug concentration time profiles, not only in the blood but in various organs or the spaces or the areas which you select. Okay students, so this was a little demonstration about a potential area of research during your M.Phill studies. Interested students can contact for further guidance and assistance at my email address, and it would be given in the description box as well.